All right, in this section, we're going to talk about how those muscle fibers are able to actually contract. So we're going to start off with how the shape and the structure of a muscle cell. So a muscle cell is also known as a muscle fiber. When the skeletal muscles are viewed underneath a microscope, they have very distinct bands called striations. That's what we sort of mentioned earlier, whether they were striated or unstriated muscles. So when they have very distinct bands called striations, those are going to be formed by the arrangement of myofibrils within the cell itself. So myofibrils are just specialized bundles of proteins, and each myofibril is going to contain two types of myofilaments. There's myosin filaments, which are going to be really, really thick, and actin filaments, which are going to be much more numerous, and those are going to be much thinner. If you remember, when we talked about cytoskeleton structure, we talked about actin and myosin filaments, or thick and thin filaments. We also talked quite a bit about intermediate filaments, which aren't going to play a role necessarily here, but they are going to be very important later on. So here is what a muscle fiber looks like. It's going to be roughly cylindrical in shape. So here's our muscle fiber. It's going to be round in shape. It's going to be grouped inside of the larger cylinder, which will be, or grouped inside of these larger cylinders or even smaller cylinders known as those myofibrils. So this is what each one of these little, striated portions are going to be, those myofibrils. Those are going to be made up of even smaller cylinders called myofilaments. So the muscle cell is actually a set of small cylinders, myofilaments, assembled into larger cylinders called myofibrils, all clustered within the largest cylinder, which in and of itself is going to be the muscle fiber. So here we have our muscle fiber broken down into myofibrils, broken down into myofilaments. All right, and so this is going to be just the words for what I just said. So if you want to take the time to write all of that down, I'll give you the opportunity to pause on this slide. All right, so we're going to look at just how these muscle fibers are able to contract. And they do this by actually causing those filaments to slide past each other. So if you look in our previous section where you'll see we have these thicker bands, these thicker, darker bands, and then we've got these lighter bands. What we're going to see is that these myofilaments are going to actually slide past one another, causing the entire length of that muscle fiber to shorten. So the myosin head, which is going to be, located on the myosin filaments, is also known as a cross bridge. This is going to attach to an actin filament nearby. When the head bends and swivels, it's going to pull the actin filament sort of past it. You can kind of think of this as like a line of sailors on the deck of a ship grabbing onto a rope. If everybody grabs that rope and pulls in one direction, that rope is going to slide past you as you're, hoisting up the anchor. So you're going to be pulling that rope past you. That actin filament is going to be sliding past the myosin filament towards the midline of the sarcomere. Sarcomere is just going to be the functional subunit of a muscle fiber, and we'll look at that in just a moment. The myosin head is going to disengage from the actin filament, and then so it's going to release, grab on, pull, release, grab on, pull. Very much like when you're pulling a rope. You grab the rope, you pull it, and then you release with one hand and grab, higher up and pull again, and then release and grab and pull, release, grab, pull. And we'll see that in our next slide. But the ability to do this, just like pulling that rope requires energy from you, the movement of the myosin heads is going to require ATP. Every little movement of that contraction of that fiber is going to require huge amounts of ATP. And it's going to move sort of in a cyclical pattern. So here's a demonstration of that. So here we have our myosin heads that are forming a cross bridge and connecting to our actin filament. So here's our actin filament, here's our myosin filament. The myosin heads form a cross bridge and they bind to that actin filament. They're going to grab onto that filament. ADP and phosphate groups are going to be bound to the myosin as the myosin head attaches to that actin. That myosin head is then going to pull back. So it's going to pull backwards and release the ADP and N phosphate. It's going to release those filaments, and then when it does so, it's actually going to release or break off from that actin filament. When it does so, another ATP binds to it. This is going to allow that allow that myosin head to return to its standard position. Once we break that ATP, it's going to latch onto the actin filament, grab that actin filament, pull that actin filament, and release. New ATP is going to bind. That ATP is going to break. When it breaks, that myosin head grabs that actin filament, binds to that actin filament, pulls that active filament, and then releases. So this is how ATP is used to funnel that system, or is used to pull those actin filaments past the myosin filaments. Muscle contraction is going to be controlled by the availability of calcium ions. So although we need ATP to cause the contraction of those filaments, or to cause the movement, the sliding of those filaments past each other, we do need a huge amount of calcium to actually talk to those muscles, tell those muscle fibers when they need to contract. So muscle contraction is going to be controlled by the availability of those calcium ions. Muscle cells contain the proteins troponin and tropomyosin, and that troponin-tropomyosin complex and calcium ions can ultimately regulate that muscle contraction at the actin-myosin binding sites. So they're going to be responsible for controlling that binding. Here's an example of how the movement of those actin-myosin filaments can actually shorten a muscle or cause a contraction. What we have is sort of a double-headed myosin band attached to these actin filaments. These actin filaments, or these cross bridges of the myosin heads, are going to grab this actin filament and pull it in towards the midline. This is what's known as the H band. It's just sort of this empty region. So these myosin heads are going to grab this actin filament and pull it towards the midline. Same with these myosin heads over here. These are going to grab these actin filaments and pull them in towards the midline. So when the muscle is fully contracted, those actin filaments are pulled all the way in. This muscle has gone from being, this long to being only this long. So we have contracted this sarcomere. We have contracted this portion of the muscle fiber. This is actually going to be happening down the entire length of the muscle fiber. Is going to be contracting in these short little bunches right here. And each time they contract, it's going to shorten the overall length of the entire muscle fiber, and it's going to shorten or contract the length of the muscle as a whole. So this is the sliding filament model. The muscle fibers contract as the sarcomeres shorten. Note that when the sarcomere contracts, so that's going to be this region here, when it contracts, the filaments themselves remain the same length. The actin filament is not any shorter. The myosin filament is not any shorter. What's happening is that they are actually sliding past each other and closing in that gap. But this myosin filament, same length it always was. This actin filament, same length it always was. The thin filaments just slide past the thick filaments. The I bands shorten, so that's going to be this region over here where we have actin filaments but no myosin. And the Z lines move inward and the H band almost disappears. So the Z line is going to be this region right here where all the actin filaments are attached. It's going to be moved inwards. This band in the center where we had myosin but no actin filaments is going to vanish. And this whole sarcomer is going to just shrink up, contract, shorten. So this is going to be done through the application of ATP. ATP is going to supply the energy for muscle contraction. The myosin filaments are going to break down that ATP, and those cross bridges are going to pull the actin filament towards the center of the sarcomere. There are several different cells that are going to be involved in muscle contraction, so it's not just about the muscle fibers, but we're also going to need our motor neurons. These are going to be type of nervous system cell that stimulates the muscle fibers to contract. We have to actually tell, like we said, the skeletal muscles are underneath our voluntary control, thus we are going to need somebody to control them, to send the message and say, hey, it's time to contract. This is going to be done with our motor neurons. Those are going to be the nervous system cell that stimulates muscle fibers to contract. A nerve is simply a group of neurons running either to or from the muscle to the rest of the nervous system. Axon is going to be the part of the neuron that stimulates a muscle fiber, and it's going to branch out so it can actually stimulate several different muscle fibers at once. So here's an example. Here is a large motor neuron that's coming down. It's going to have these axons that are going to branch out, and so this one neuron can stimulate multiple muscle fibers all in one go. All right, and that's going to be it for this section. You should be able to explain the role of the myofibril, myofilament, and sarcomere in the muscle fiber, explain how those thick and thin filaments interact in that sliding filament model. And although we didn't really spend much time on it, you should be able to describe the role of ATP, which we did spend quite a bit of time on in that muscle contraction, but also make sure you review calcium ions and their role that they play in muscle contraction.